Everything is set up.
No problem. Transmissão começou, Isa?
Começou, pode começar, professor.
Bom dia a todos e a todas. Quero agradecer a presença de todos no nosso evento. É um evento especial para o nosso grupo, para o programa de pós-graduação, o novo programa de pós-graduação em Ciências de Movimento. Esse evento é fruto do edital CapSprint, no qual o antigo programa da fisioterapia participou, um programa de internacionalização, no qual a ideia era repatriar brasileiros que estavam em outros países. Então, a doutora Luciane Magri, ela realizou um pós-doutorado durante 2 anos em Montreal, com supervisão do professor Jean-Marc. E amanhã teremos a palestra da professora Ana Maria Teixeira, no qual nós vamos explicar melhor, né, que a Luciele, né, tem uma dupla Luciane e Luciele, e ela fez todo o seu doutorado e o pós-doutorado em Coimbra, e nós repatriamos através desse edital. E para finalizar esse momento, nós estamos realizando um evento em caráter internacional, com os 2 supervisores no exterior, das 2 doutoras, com a palestra extremamente atual, né, o professor Jean-Marc, a Luciane vai apresentar todo o vasto currículo dele, mas é uma pessoa muito especial na minha formação, na formação do professor José César, que está aqui, que é professor do ICB da USP, no qual ele participou da nossa formação desde o mestrado até o cenário atual, tá bom? A partir de agora, todas as falas vão ser faladas, elas vão ser em inglês, por conta do idioma do professor Jean-Marc. Nós tínhamos 2 opções, ou inglês ou francês, né? O inglês, ele venceu, então toda a nossa transmissão agora vai ser em inglês. Jean-Marc, now we will speak just in English, okay?
Yeah, okay.
Thanks very much, because this moment is very special for me. And Luciane, now you present you. Luciane.
Hello, everyone. Hello, Jean-Marc too. And now we're gonna start your presentation. I would like to say that I'm super happy and excited to have you here, because everyone who knows you knows that you are a great professor, and it's a pleasure to have to make this bridge between your knowledge and bring your knowledge to all of you here in Brazil. Of course, that we would like to have you here physically, but of course our condition right now it's not possible. But I would like also to say that Jean-Marc is not just a great professor, he's a great friend, he's a really good human to share not just his knowledge, but a lot of experience. And that's what I would like, that all of you have this kind of experience during this presentation that he gonna do right now. So Jean-Marc, it's all yours, and you can start your presentation and say, feel free to start your presentation, right?
Okay. Well, obrigado, Luciane and Fabio. It's a real pleasure to be with you this morning. It would be better to be physically present, but at least we're together. So today I'm going to talk to you. I'll try to speak slow because I know some of you are not that familiar with English all the time. If I go too fast, you let me know, okay, Lucian?
Okay.
Now, I'm going to talk about bone, skeletal muscle crosstalk during exercise. I would like you to know that I'm not an expert in this sort of things, but I've been doing some reading and I found things very interesting. So I want to share it with you. So you go easy on me on tough question after, because this is not really my field of expertise. Okay. Maybe 30 years ago, or maybe even more, the only way that we looked at how metabolism is regulated during exercise was through the central nervous system. For instance, I work on for a little while on communication between the liver and the pancreas. And the way we look at it was that they were afferent nerve going from the liver to the brain, and then efferent nerve going to the brain to the pancreas. So the liver was talking to the pancreas, but everything went through the brain. But as years went by, people realized that this concept was not perfect, and there was probably way to one organ to talk to the others by doing something else than going through the central nervous systems. Well, the first step in understanding the crosstalk between organs was the finding of adipokine that I'm sure that all of you have been known about. So they have adipokine, and those adipokine are molecules coming from the adipocyte. Well, that was the first finding. And then as the years went by, we discovered that they were also myokine. So some molecule coming out of the muscle and doing something, going to other organs and doing something to regulate during rest and also during exercise. Among those myokine, you find irisin, interleukin-6, and so on. So as I got interested a little more about these, what they call exercise-inducible factor, I look at the liver, and we found out that they were also hepatokines. And well, I was starting to read a little more about hepatokines since I had some time free since I'm retired now. So I've been some more reading about hepatokine, like phytwin, that Lucian knows very well. And then I was getting informed about those exercise-inducible factor, I found out that they were also osteocalcin. Well, that was interesting. And then I got more interested in what's going on with these osteocalcin. But there's a lot of those molecules now that comes from almost every tissue going to other tissue, and they have a role to play. They establish inter-organ crosstalk, and that is a very new way to look at regulation during exercise. If you are interested, there's a reference that I suggest to you. This reference is a review paper, very recently, 2020, a review paper where you find the role of exercise with myokine, hepatokine, osteocalcin, and adipokine. So that's a good way to start your reading, is if you want to get a little more knowledge about these crosstalk. But today, I'm going to talk to you a little more about bone and muscle. Well, the first question that we could ask ourselves is, why would bone talk to muscle, first of all? Well, first, bone can sense mechanical forces. So there are some way of bone are not inert, they are very much alive. So they can sense mechanical forces. They are very close to the muscle, of course. So they are both involved in the same thing, when they're put into action. And also, and most importantly, the fact that exercise capacity and bone mass both declined at the same time. So there were some very good epidemiological studies showing that decline in exercise capacity and decline in bone mass happen at the same time. So people start looking a little more about what's going on in the bone. And people find out that they were, as in muscle or adipocyte, they were osteocalcin. They call it osteocalcin. Not so long ago, 2007, maybe this, I'm not too sure about this date, maybe a little more, but it's very recent that the people found that there were some molecule coming out of the bone, and they call it osteocalcin. There was some sort of bone-derived hormones, if you wish, and mostly the two of them, the most important of the two of them are osteocalcin and lipocalin. So what do they do? Well, what do they do? What do these molecules do in the body? Well, osteocalcin. First, it was involved. That was found from not that recent study, nothing to do with exercise. He found that osteocalcin is involved in memory and also in male fertility. Well, I'll come back to male fertility a little later on because it's important, male fertility. But also, they found that osteocalcin stimulate the production of insulin. That was known for a certain number of years, but also energy expenditure, glucose homeostasis. So there were some involvement of osteocalcin in some sort of metabolic regulation. That trigger, the interest to look at this osteocalcin during exercise. The other molecule coming out of the liver is of the bone. Is lipocalin. Lipocalin. What we know about lipocalin is it influences energy metabolisms by activating appetite suppressing signaling in the brain. So it has an effect of regulating appetite. I found that very interesting that an hormone coming out of the bone have a way to regulate appetite during life. So, but I'm not going to talk about lipocalin today. I'll focus on osteocalcin. So, on the in summary, you have osteocalcin coming somehow out of the bone, and it has an effect on insulin and insulin sensitivity, which we're not going to talk that much about today. It has also an effect on testosterone secretion. So it's interesting because osteocalcin has been known to increase testosterone secretion. Osteoblasts, through production of osteocalcin, increase testosterone production, but they have no effect on estrogen production. So nothing to do with ovaries, but it has an effect on testosterone. That's an interesting field, that someone might have to might want to study a little more. What we're interested in today is osteocalcin on muscle. So it has an effect on muscle, nutrient uptake and catabolisms, and I will talk to that a little more with that later on. And osteocalcin has also an effect on cognitive function, and as lipocalin has an effect on appetite through food intake. So today we're going to focus a little more on what's the effect of osteocalcin on muscle, and does have something to do with exercise. So let's talk a little about osteocalcin, just to see, to know a little more about osteocalcin. It's synthesized by the osteoblast. In the bone, you have different type of cells. You have osteoblasts, which are the main cell that incorporate calcium into the bone, and those osteoblasts become osteocytes when they're very much calcified inside of the bone. It's a continuum. But the most, also with the osteoblast, you have osteoclast. And the osteoclast, they do a little the inverse of osteoblast. So they do decalcification. So you have a process going on all the time in the bone that is called bone resorption as part of the process of bone remodeling. So the bone is all the time remodeling, with osteoblasts incorporating calcium and osteoclasts taking out calcium. And the two of them work together. As far as osteocalcin is concerned, osteoblasts are the cells that synthesize osteocalcin. But osteocalcin coming out of osteoblasts are not active. they have to go to osteoclast, and then there is a cleavage, the molecule is cut down, and then osteocalcin becomes bioactive. So you need these two type of cells to have osteocalcin, and it's important to realize that there is a very active remodeling process going on all the time. What we know also about osteocalcin, it's a very short half-life in human, at least. In mice, it's different. I'll talk to that about that a little later. It has a short life, so you cannot, have a lot of osteocalcin all the time. And also, it greatly declines with age, as bone mass declines with age. Now, what are the physiological action of this osteocalcin? Well, as you may guess now, what we knew from the older study is that there is a very good correlation between osteocalcin and the basal metabolic index, visceral fat mass, CRP, which is a molecule coming out of the liver, and it's a good reflection of inflammation in the body. So it has something to do with good health condition in the body. And also it has an effect on insulin secretion, as we talked about a little earlier, increased beta cell survival and insulin secretion. So those physiological action are interesting in the context of exercise. So eventually people look at exercise. So what's going on during exercise with osteocalcin? So people thought that the exercise might be an interesting physiological action to study osteocalcin. Well, what they found first is, does osteocalcin levels change during exercise? And it does. Osteocalcin level double during exercise, but in young adult mice, 3 month or 6 month old, you can find and double the level of osteocalcin. But it does not increase during exercise in older mice. If they are older, no osteocalcin coming out. So that means that already the bone is not doing what it's supposed to do. Now, the other way to look at osteocalcin during exercise is to inject osteocalcin. Well, this has been done, in some study. A single injection of osteocalcin, whether before exercise or maybe a chronic delivery with a mini pump all the time to maintain the levels, or just osteocalcin secreted before exercise, the result is you increase exercise capacity of young mice to about 20%. Exercise capacity here is the longer of time that a mice can run on the treadmill at a determined speed and slope. So they measure exercise capacity. Several studies found an increase about 20%. But most interestingly, if you inject osteocalcin before exercise, you restore the exercise capacity of older mice, 15-month-old, to the level of young adult mice, 3-month-old. Well, that's very interesting, because if you have osteocalcin, then in some way you sort of compensate for the age-related problems, at least as far as exercise capacity in those mice are concerned. Now, the other way to look at the effect of osteocalcin during exercise is to knock out osteocalcin expression, or to block the receptor of osteocalcin. Osteocalcin has a G protein-related receptors, so you can find a way to block the expression of this molecule. So whether you don't have osteocalcin or whether you don't have the receptor, if you do that, one or the other, the knockout mice ran about 20 to 30% less time than control litter mate. And also there is a decrease in maximal oxygen consumption. So it shows that, at least from these studies, that osteocalcin has an effect that reduce exercise capacity or increase exercise capacity if you inject this molecule. What about the receptor? Let's just say a word about the receptor, the G protein receptor of osteocalcin. Well, what we know about this receptor is that this receptor, the osteocalcin receptor, is more expressed in skeletal muscle than in most tissue. So the link between osteocalcin and the muscle is very strong. It seems to be more in oxidative than in glycolytic muscle. And if you block osteocalcin expression, you found that in muscle, the expression of the G protein receptor was 3 times higher. So this is a sort of a compensatory. The muscle doesn't see the substrate, and it increases the expression of the receptor to get the effect. And also, a single injection of osteocalcin increase the. I don't see. Okay, sorry. Oh boy, what did I do? Oh, Lucian, you still there? Oh my
Yes,
God.
I'm here. You can click again in your
At the end?
presentation mode. Yeah.
Click on my presentation.
Yeah.
That's okay.
And put to
presentation
present.
mode. You know, there is something at the end.
Yeah.
There's something at the.
I can't see it's writing here, but I think that's possible to think of this.
Should I get rid of this?
Yeah, I think that's possible.
Hide.
Okay. I'll go without. I'll forget about this. You know.
Okay.
Okay. Let's forget about this. Well, you say masque, maybe. There you go. All right. So finally, about the osteocalcin receptor, a single injection of osteocalcin increase the exercise capacity if you don't have osteocalcin, but if you don't have the receptor, doesn't change anything. So the receptor it shows that this receptor is really specific to osteocalcin, and it has an effect, a strong effect on the muscle. So what does osteocalcin do exactly in muscle? At least, you know, at rest and also eventually during exercise. Well, what they found is that osteocalcin does not affect mitochondrial number and function. So you don't affect the machinery such, you know. What it does is it promotes directly the uptake and catabolism of glucose, including glycogen breakdown, in skeletal muscle. So it affects, for instance, translocation of GLUT4. It affects the phosphorylation of AMP kinase, which is a kinase that is very important during exercise. I'm sure you are aware of that. So what the osteocalcin do exactly is it favors the entrance and the utilization of glucose during exercise. And also, it favors lipid utilization during exercise. So, for instance, if you block osteocalcin, if you block the receptors of osteocalcin, AMP kinase phosphorylation or activation is reduced. Conversely, exogenous osteocalcin increase AMP phosphorylation. The other way to look at lipid utilization during exercise to try to demonstrate that lipid utilization is affected by insulin is if you block the receptors. If you block the receptor, what you found is an increase in malonyl-CoA in the cells. Well, malonyl-CoA, as you probably know, block CPT and block entrance of lipid inside the mitochondria and then reduce, of course, utilization. So if you have a lot of malonyl-CoA, you're more on the way to synthesize lipid than on the way to utilize it. So these things sort of demonstrate that osteocalcin has a profound effect on glucose, glycogen, and also on lipid utilization. Finally, the expression of transporter for fatty acids, CD36, FAT-P1, and also CPT1. They were all reduced. The expression were all reduced if you block the receptor of osteocalcin. So osteocalcin has an effect on glucose metabolisms and glycogen metabolisms, but also on lipid utilization. So now I hope I sort of convince you that osteocalcin has an effect on muscle. But if you want to talk about feedback, we have to, the next question would be, how does muscle talk back to the bone? Otherwise, you don't have a crosstalk, So does muscle talk back to the bone? Well, in addressing this question, the key word is interleukin-6. Osteocalcin established some sort of a feedback loop to the bone via interleukin-6. So let's talk a little about interleukin-6. I'm sure that a lot of you know a lot about interleukin-6 because you've been working in immunology with Fabio and so on. What we know about interleukin-6 rapidly, interleukin-6 is secreted by adipocyte and monocyte and is classically considered as a pro-inflammatory. See, it's a pro-inflammatory molecule like TNF-alpha, CRP. But when released in large amount, the people working in Scandinavia, they show, it's very well known now, they show that during exercise, interleukin-6 coming out in a very large amount out of the muscle. For instance, it can increase by 5-fold after 30 minutes of running, up to 100-fold during a marathon. These have been documented data. And it is believed, and there is good evidence for that interleukin-6 with these in large amount exerts an anti-inflammatory action and also an insulin sensitizing action. So this is what we know most of the time about interleukin-6 during exercise. And then the people start asking the question, is interleukin-6 have other physiological effect during exercise? Well, they found a lot of things. During exercise, they found, for instance, that interleukin-6 coming out of the muscle seems to be crucial for the loss of visceral adipose tissue. So it has something to do with utilization, lipid utilization during exercise and so on. It directly increased GLP-1. GLP-1 is coming from the enterocyte and has something to do with insulin secretion. So it's a good point for that. Stimulate lipolysis in white adipose tissue that goes with visceral adipose tissue. And also it promotes glucose uptake and free fatty acid oxidation in skeletal muscle. So the point is that interleukin-6 coming in large amount out of the muscle during exercise is not only anti-inflammatory, but it also have a physiological action that is good for exercise, that contribute to proper regulation during exercise. Next question then is, what stimulates interleukin-6 production by muscle? Well, this question is a little more difficult to answer. And when people don't know exactly the answer, what's going on, something going on in the muscle, well, they assume that it's probably skeletal muscle contraction. It could be calcium that trigger this synthesis of interleukin-6. Could be inflammation. Well, we don't know exactly. But what the people interested in osteocalcin found is that osteocalcin is necessary for the increase in interleukin-6 in skeletal muscle. So now here they use the word in those studies that you would find at the end of this presentation, by the way. Those researchers, they use the word necessary. That means you need osteocalcin to increase interleukin-6 in skeletal muscle. Well, that is a very strong statement. So what's going on? If osteocalcin stimulate interleukin-6, then what about the crosstalk? So what they found? Well, it's been shown that interleukin-6, in addition to have physiological effect, also signal to osteoblasts and favor osteoclastogenesis. Osteoclastogenesis is synthesis of new osteoclasts. And you will remember that osteoclasts are needed to activate osteocalcin. So osteocalcin seems to signal to the bone. These things have been shown in, I think it's culture cells. When you block osteocalcin, when you block interleukin 6, by blocking the receptor, when you shut down the receptor in cell that have been cultured with interleukin 6 and osteoblast progenitor cells, if you block osteocalcin, then the number of osteoclasts and osteoblasts are greatly reduced. So it shows, these studies, these approaches show that interleukin 6 seems to be necessary to stimulate osteocalcin secretion in osteoclasts and osteoblasts. So, what's going on during exercise? So what's the concept during exercise? What are the crosstalk between bone and muscle during exercise? Well, you have to first understand that exercise induces bone resorption. These mechanisms that I talked to you at the beginning of the presentation, exercise induces bone resorption. So you have calcification, decalcification. So it's increased during exercise. And therefore, bioactive osteocalcin is increased. So that creates a sort of feed-forward loop. So you have more osteocalcin, as we showed before, and it's because of bone resorption. So what's going on during exercise in this crosstalk? Well, you have more of osteocalcin that are synthesized and released by the bone due to at least bone resorption that is increased. This bioactive osteocalcin goes to the muscle where there is a receptor for osteocalcin in the muscle. And what happens here is you favor nutrient uptake and catabolism, that means utilization. So you help the muscle with that. It seems to be necessary. But in addition, osteocalcin will favor the synthesis of interleukin 6. And interleukin 6 will be released from muscle in very large amount. And this interleukin 6 has different action. It goes directly feedback to the muscle and help also contribute to nutrient utilization. It has some effect on liver with glucose production, free fatty acid production. But also, it feeds back to the bone. Well, there is interleukin receptor, and this interleukin 6 would favor even more the secretion of osteocalcin. So you establish some sort of a loop between bone and muscle through osteocalcin and through interleukin 6 most likely during exercise. So here's some other data supporting this crosstalk. Some of them. For instance, if you, in the muscle, you block the receptor of osteocalcin, the gene whose expression is the most decreased after exercise is the one encoding interleukin 6. No other myokine known to influence exercise were affected by the absence of osteocalcin. So osteocalcin and interleukin 6 in muscle are very much related, according to these data. More convincingly, the rise in circulating interleukin 6 induced by exercise was blunt if you block the receptor of osteocalcin or if you block osteocalcin. No osteocalcin or no action of osteocalcin on the muscle, interleukin 6 level are very much reduced. So it shows, that osteocalcin is very important for this action. However, well, this is not that important, but just mention it, osteocalcin does not require the presence of interleukin 6 in muscle to act. If you infuse osteocalcin, even if you don't have interleukin 6, you will have an effect of osteocalcin in muscle. You don't need the presence of interleukin 6 for osteocalcins to act on muscle, but you need interleukin 6 to stimulate osteocalcin eventually. All right. All those studies altogether, those studies were done in mice most of the time or in culture cells. But it's interesting to say a word at least of what's going on in human. it's a little more complicated to study, of course. In human, what we know, if you try to maintain an increase in circulating osteocalcin for 2 days by infusing osteocalcin, as you know, osteocalcin has a very short life in human. But if you do that, you would increase, you will find, oh, I'm sorry. Oh, okay, let me start all over again. If you train people, some people, non-diabetic human subjects, have been trained for 12 weeks, and what they found is an increase in circulating osteocalcin, a 60% increase in osteocalcin. So people that are trained or well-trained, they may find an increase in osteocalcin in the level, which is, of course, a positive action. If you the same exercising obese subject, if you block the receptor, it's that can be done in human through this antibody. If you block interleukin-6 receptor, the increase in circulating osteocalcin level by exercise virtually disappear. So you need this interleukin-6 to have the osteocalcin secretion during exercise in human. So those are this, very few study that has been conducted in human, but I guess this is a field that is going to be expanded in the next years. So in summary, interleukin-6, some way of summary, interleukin-6 signaling in osteoblast is needed to enhance exercise capacity during endurance exercise. The studies I'm referring to use the word needed, is needed. So it's very important, Mice liking interleukin-6 receptor only in osteoblast, exhibit a deficit in exercise capacity. So these feed forward between bone and muscle are very interesting during exercise. So overall, what you have? Well, you have, the bone, the osteoblast, and you have the osteoclast, and they communicate one another. And osteocalcin secreted from this osteoblast go to osteoclast where they cleave it. And then you have the osteocalcin that is bioactive. This osteocalcin as such would favor exercise, long endurance exercise, mostly by increasing nutrient entrance and utilization in muscle. But this osteocalcin, active osteocalcin, will go through the muscle as well, and through the receptor will favor a lot of things, physiological action, but also would favor the release of interleukin-6, important release of which feed back to the bone, which has a contributing physiological action as such, but would feed back to the bone, and then you increase the feed-forward loop that is seems to be so important during exercise. Now, 3 and 6 month old mice, where interleukin-6 receptor was blocked, they display a severe decrease in their ability to perform endurance exercise. If you don't have the interleukin-6, you decrease performing exercise. So it seems that interleukin-6 is needed to signal to osteoblast to maintain exercise capacity. Oh, that's very interesting as far as I know. So in conclusion, well, if you want to come back, and have some sort of the feed-forward condition, we can ask ourselves if injection of osteocalcin might be a way to prevent the age-related decrease in exercise capacity, or even the age-related effect during exercise. Well, this question is interesting in human, but the problem is that osteocalcin has a short life in human. But I know that some people in Montreal at the Clinical Research Institute here in Montreal, they work on try to synthesize the same type of osteocalcin that they found in mice. And the difference is that there are a few carbohydrates attached to the molecule. And if you attach those carbohydrates to the molecule, then you prolong the life of the molecule, of osteocalcin molecule. So they're sort of trying to find a way to have to inject osteocalcin in human and maybe decrease exercise capacity or decrease a lot of things that's coming on with exercise. So thank you for your obrigado for your attention. And those are the two studies that you might want to look at if you wish to learn a little more about this bone muscle crosstalk. And those are the studies that I use, or some others, but those are the main two studies that I use to prepare this presentation. Obrigado.
Obrigada. Thank you so much, Jean-Marc. It's a really good presentation. And just to let you know, while you are presenting, I understand that for you may be kind of lonely, but we have more than 100 students watching your presentation. So it's super nice and a great presentation. And while you were presenting, we had some questions that I am gonna put here on the screen for everyone in YouTube. And the first one is from Igor Massari, and his doubt is about if osteocalcin has more receptor in oxidative fibers, and if it's possible to optimize their using during aerobic exercise. If it's possible to increase the receptors from osteocalcin in muscles.
Yeah, well, I would answer the question as far as I know. And as I said at the beginning, I'm not really an expert in this bone-muscle relationship. That is very interesting. I found it very interesting. It's brand new, at least to me. Yes, the first thing is it is more present in oxidative than in glycolytic fibers. So the effect of osteocalcin injection is more prominent in long endurance exercise than in short or very intense exercise. That's one thing. Is there a way to increase the receptors? The only way that I can see right now is through training. And through training, they found that first there would be more osteocalcin in the blood first. And I don't know if they measure number of receptors on the cells, but from what we know from physiological action, I would guess that they are more receptors. Is there a way to increase receptor and osteocalcin except training? You mean by doping somehow?
Yeah.
I don't know, but there must be a way. There must be a way. People always find a way. But well, yes, you could inject osteocalcin, and I don't know if it's it's forbidden or something. I really don't know. But I don't know if it's known, But it's interesting to see. I was a bit surprised by the words that these people use, like it's absolutely necessary. Osteocalcin is absolutely necessary. Those are strong words, and I find it because exercise regulation is complex. It's not only one thing or the other, but it seems that, still has to be checked in human because, exercise capacity in mice on the treadmill, they measure the time that the mice exercise. But as you know, and probably you've done that before, or Fabio has done that before, the timing of how long the mice run on exercise, there is some subjective way in this, do you put a lot of shock or, this is not perfect, this is not perfect. It's an indication. I would say I would not have used the word necessary or absolutely needed, but there is a link and there is something going on. It's interesting, very interesting, and it shows to me. It's a long answer, right? It shows to me that if you exercise, if you mobilize yourself through life, then you mobilize the bone. It's not only good for the bone, to sort of avoid problem with the bone, like, women, they lose the bone as they get a little older. But it's good for the metabolism. It's good for insulin secretion. It's good for diabetes, I guess. So the point here is that putting the bone into action, it's interesting for your health and exercise capacity. To put the bone into action, you cannot just swim, because when you swim, you don't put your bone into action really. You have to stand up and put some pressure on the bone. So it's interesting, exercise doing is great. We all know that. You and I, we all know that. But you have to put some pressure sometime on your bone, and that's my long answer.
But this view of something inducing IL-6.
Sorry, Marilia have two questions.
Oh no, not Marilia. Question, I guess.
Yeah.
I knew it. I knew she would come in asking tough questions. She always does.
So first, Jean-Marc, it's so incredible to see you here. And
Yeah.
I mean, you have not changed it at all. You look
I have
all
not.
the same.
I lost a few hair though.
No, it's spectacular, just the same. So I'm very glad to see you.
See.
And thank you for this very stimulating talk. So, I still work with the cachexia thing.
Yeah.
And I was wondering, I have two questions actually. So the first one concerns satellite cells. Do you think osteocalcin,
What kind of cell?
satellite, muscle
Oh, satellite cell. Oh, in a muscle, right.
Yes. Do you know if osteocalcin has any effects on satellite cells? Because
Yeah, from what I read, Marilia, I read maybe, I would say around 15 to 20 papers around this field, and I've never seen the word satellite cell.
right.
I don't know. There was something to do. Yeah, I don't know. If
No,
you would think if osteocalcin would stimulate satellite cells, yeah, that's a good, interesting question.
Yeah, because with the patients, we have these problems with the satellite cells, which are dividing, but they are not fusing. So maybe it's a metabolism thing. And I thought that maybe this could be very interesting to have a look. And if you're willing, we could have a look at these together in the patients. We also have exercise patients, cachetic and exercise. So we could look at this.
If you want to have a look at this, I would let that to you or Fabio or Lucian, because I'm retired now.
Yeah, well, but you can always give your input. And the other thing
I'm
is
retired now. I just hang around, and I don't do no more research.
and the second thing is there's another hormone secreted by the muscle that I think I learned about it last year when I was in Germany. It's periostin. So have you heard about this periostin thing?
Periostin thing?
Yes. Is it related?
That's an hormone or a molecule coming from the outside of the bone, the periostin?
Yes, because maybe one interesting thing to have a look at is the ratio between periostin and osteocalcin.
Calcin.
Yes, because
Yeah. Maria, to be honest, I don't know much about the bone, never worked with the bone.
I know nothing.
For all those years, I know never really, I don't know much about except there are a few cells, osteoblasts, osteoclasts, osteocytes, basic stuff. But that's why, I'm not really an expert in bones. So periostin, no, I haven't, those from my readings, I haven't seen that. That's interesting.
Okay. Thank you very much. So I'll let the students ask, because otherwise I will monopolize, and I miss you so much. So I wanted to speak. I can do that later. So.
Right. Nice talking to you. Yeah. You still look great.
Not ready.
Okay.
The Professor Romulo first.
Oh yeah.
Professor Romulo, can you speak?
Yeah, it's difference between the effect of both things. Well, I had the question. I could read the question. It was in English, but then it disappeared. I was not quite
yes,
finished.
yes. Just a moment.
Put back the question. You put back the question.
Moment. I'm trying to put on the screen. Just.
I think the question was if the osteocalcin they inject in all mice that makes them exercise longer. Where was it carbonated or not? Yes, it was. You know, the influence a role. Oh, how does the anaerobic? No, that's not the question. Let me try to answer the previous question.
The previous?
Yeah, I guess that the osteocalcin they inject was the mice osteocalcin. That means it was carboxylated. Now, if it wasn't carboxylated, I don't think it would be as effective in mice as it is in human. So I guess it was carboxylated, and that's it. The positive effect of an old mice by injection of osteocalcin is through carboxylated or uncarboxylated. Is there a difference between the effect on both? Well, there must be an effect, you know, because carboxylated is specific to the mice, and non-carboxylated is specific to human. I don't know about other type of animals. I don't know. So I guess it was carboxylated. Otherwise, it wouldn't be specific to the mice. It has to be. It wouldn't be as much active. You know. I guess that's my simple answer. Yeah. Next question.
From Romulo. Romulo, could you let's open your screen? No.
Open my. You mean the PowerPoint?
No. It's just Professor Romulo, because I.
Oh, you talked to someone else?
Oh. Yeah.
Or the Ricardo can other question also. The Ricardo is Romulo's student can.
Okay, good. I'm opening.
Yes. Firstly, Professor John Mark, congratulations and thank you for your presentation. It's really good for me, especially because our subject of study is to understand the effect of exercise on bone health in children and adolescents. Was really good for us to improve our knowledge in the subject. Thank you very much, Professor. And the first question of Professor Romulo is if the relationship between osteocalcin and IL-6 during the exercise could be different in pediatric populations compared with adults.
Could it be different? Well, you know, I do not have any data, you know, to support my answer. Could it be different? Is interleukin-6 higher in young children during exercise? Fabio,
Yeah.
you know? Is it? I don't know. I would say it is probably different, you know, for children because of the puberty, you know, and everything going on at the same time, you know, so probably around 13, 14, 15 years old, it would be difficult. For younger children, I don't know, but I really, the answer is I really don't know. I don't have any data, but I think it has to be looked at because it would be a little different, you know, probably because everything's a little different at that time because of the growth of the children. Things change a lot, you know, from one year to another. So it's very difficult. I guess it would probably be different. You know, exercise capacity is not that high before puberty, and then so it would be different, I guess. You know, probably it's a working process. You know, you don't know. It's a developing process.
Thank you, Professor. I think it's good because if the work is going on, you can try to do some research about the subject, Professor.
Yeah, sure. Yeah, it's really a new field, you know. I guess, you know, it's very new. The review that I started with is 2020, so it's
pretty new.
Yeah, it's very to me at least. But so I think I wasn't that bad, you know. So I am following the stuff. So it's very interesting. Very interesting.
Thank you, Professor. My second question is about the effect, because we know that IL-6 from the inflammatory disease or chronic inflammation, they can have like a increase, bone remodulate, and this could be worse could be harmful for the bone. In this case, the IL-6 from the
Could be
muscle
bad for
have
the
a
bone?
different effect.
I'm sorry again. I missed your last part.
That is fine, Professor. My question is, we know that IL-6 from systematic inflammation or some disease can be harmful for the bone. In this case, I can think that the IL-6 from muscle have a different receptors, and these receptors is more useful for bone formation, not for bone remodulation, like we have in disease, rank L and another receptors on osteoclast.
Or again, you know more about bone than I do, So
No, I don't.
you're more able to answer the question yourself, A different type of receptors? I don't know. I really don't know. I'm sorry.
No, thank you, Professor, because I was thinking about, and maybe the place where this IL-6 is from is very important to understand the effect on bone.
Mm-hmm. Yeah, probably has to be looked at a little more specifically, but right now I don't even can speculate about this. I'm sorry. You want to talk about liver? I know a little more about liver, but
yeah, liver.
It was really good for me. Thank you.
That's the risk of making a presentation that you're,
another field.
I've done the presentation for you, but I've done it for me too, to learn. So we're both all learning together.
Yes, I learned a lot with your presentation. Thank you, Professor. Thank you, Professor Fab and Professor Lucien, for the opportunity.
And Ricardo, stay here, please, because I have one question, one discussion, because Jean-Marc explained about the exercise, but the endurance exercise. And Ricardo studied, published in last year, one medicine science about the different between different sports, the swims and the basketball players, the football players, is it the different bone density, the bone mineral density. And one good question is, this different bone mineral density can induce the different osteoclasts and IL-6. Is one good question, Ricardo. Yep. Can you explain about?
Yes, sure, Professor, I can. Professor Jean-Marc, in this paper, we compare 10 different sports modalities. We compare the bone mineral density of 10 different sports modalities among adolescents. And we saw that if you're considering like a volumetric interpretation of bone, the gymnastics is the best sport. It makes sense because gymnastics will have a lot of impact on bone during the movements and the jumps. And the second one is football, track and field have a similar effect as well. But we didn't check if the sports can affect this IL-6 and the osteocalcin of these modalities. Maybe these factors could help to understand and explain, not just the impact on the ground, maybe this effect of osteocalcin, because it's very important to these variables, and maybe the IL-6 from the exercise, not from the systematic.
Did you measure osteocalcin in this study?
No, in this big sample, I don't have this information, unfortunately. But maybe we want to start to considering now osteocalcin, CTX, and P1 and P as well, to try to understand this different effect on bone.
Oh, good. Yeah.
Yeah, I think that can no change the osteocalcin or IL-6, but can change the receptor, the osteocalcin. And we
GPR receptors. Yeah, sure.
Yeah.
Maybe.
It has to be looked at. Interesting.
Because
we could. Well, it's interesting to look at different type of activities. You could wonder if you increase bone solicitation just by jumping, you increase the bone, would that have an effect on interleukin-6 and on nutrient regulation eventually? I don't know. But point is, the health of the bone is important, not only for the bone, but for the body. It's very interesting. It's the same thing everywhere, everywhere. It's I wrote a book, that I gave to Luciane, but she didn't read it because it's in French. But in that book, the point is that
I need to say that this book was in French. I have to say this.
But in this, I try to defend the position that every organs contribute to the whole body. Everyone contribute, and if you don't put into action those, you're missing something. The body has to move all the time, not just for one thing, for everything. So it's an entity, where everybody is communicating with everybody. Well, everyone is communicating with everyone all the time, So that's it's interesting, very interesting. It's a very new approach of looking at regulations during exercise, but also I would say the effect of exercise on the health, on general health. Not just pancreas, everything, the brain. Today, you find papers published with the effect of exercise on everything: the brain, Alzheimer, the liver, the enterocyte. Now it's the microbiota in the intestine. Oh, it's very important now in everything. So it's this crosstalk was very, very, very important and very interesting. A little complicated. It gives work to you folks to work for years.
Yes, Professor, thank you again for the opportunity. It was a pleasure and an honor to meet you. Thank you again, Professor.
Yeah, nice talking to you. I knew I was going to find someone who knows bone and everything, though.
No, but I learned a lot, honestly. I really appreciate your speech.
It's very superficial. It's not, I didn't show any data, but, well, it's the first thing. Good.
Thank you.
My pleasure.
That's good.
Zeka?
So,
no more question?
Zeka, I have one question.
You have a lot of questions, Jean-Marc.
Zaka has a question.
Let's manage Zeka here.
Thank you for this great presentation about this hot topic, the kines, the osteocalcins and the adipokines and the kines, etc. Yes, but my question is about the effects of osteocalcin is paracrine or endocrine. This we observe this increase of osteocalcin during exercising in serum because I think that this potential role of osteocalcin in fatty acid metabolism is very interesting because the monocytes and macrophages probably have a great amount of these receptors of osteocalcin.
Really?
And I think that it's possible to understand the link between the M2 alternative activation of macrophages because the osteocalcin. Do you know if is increase the osteocalcin serum?
I really don't know, Zaka. I wish I could give you a good answer, but it's interesting. What you say is that osteocalcin might also have an effect on macrophage or these immunology cells, and increase immunology protection or something, Well, it's very well possible. It's very well possible. I wish I could answer the question, but you check that out and you let me know. It's a good thing. It's a good point. Sorry, I can't go any further.
Thank you.
Yeah, it's interesting this view that because normally when we study the effects of muscle contraction. And we already understand the metabolic effects of IL-6 and everything, but something that stimulates IL-6 in the muscle, it's a new field, at least for my point, like because we, as you mentioned, Jean-Marc, we were used to study liver metabolism, so understand something that stimulates IL-6, that's super metabolic interleucin. So it's interesting view to be discussed.
Yeah.
And
we
also,
now, what's the effect of osteocalcin on the liver?
Exactly, on the liver. But fettuine is interesting too. No, fettuine.
Some sort of effect, you know, osteocalcin on the liver.
Jean-Marc,
I would... and then you know, you have an effect of osteocalcin to reduce the age-related effects. You know, the effect of aging, and you have to find it fast because I'm not getting any younger, so I need the answer.
Yeah.
You know, I need the answer fast.
Yeah,
so get to work. You know, don't stay home. Go to work.
To be younger.
Yeah, stay younger as much as we can.
Yes.
So Zaka, how old are you now, Zaka?
What?
What's your age now? How old are you?
What? I don't.
I'm asking.
How old?
Quantos anos? How old are you?
38.
40, yeah? 40,
yes.
Oh, my God.
40.
I've known him. He was a small boy, you know.
Yes.
That's a long time ago, yeah? Boy, it's a long time ago. Oh,
yes.
Times go fast. Unbelievable.
We need to start to think about.
You in Brazil, you stay young all the time. You know, we know that in Brazil, everyone is stay young. It's a young country. A lot of people are very young. You know, they say that for a while, 75% of Brazilians, they were under 30, 35 years old, something like this.
Yeah.
Yes.
It's amazing. Here's a bunch of old folks, you know.
Jean-Marc, I'm putting on the screen. Of course, I believe that we already discussed, but it's just to keep including the postgraduate students in this discussion. Annelise, she asked, how does exercise intensity can influence the role between osteocalcin and IL-6 performance?
I think I do not have a precise answer to this question. I don't have data, but my experience is telling me that exercise intensity would solicitate bone even more, and then would stimulate osteocalcin synthesis and liberation. It would increase this forward loop. One thing is that, interestingly though, in the readings that I've done, they did not find any change really in muscle mass. You know, they did not find hypertrophy or that sort of thing. But they were using mice, and sometimes in animals. In animals, they don't hypertrophy as well as human, you know. And they were using only endurance exercise, but did not find a muscle mass change. And I would not think it would change muscle mass. It would have an effect on nutrient, but I think exercise intensity would have an influence.
Yeah.
I would try to combine both, I guess.
Yeah.
Good question.
And
good question.
We did this one. I didn't read yet. It's the other one from.
Does the anaerobic can influence the role of osteocalcin? Can muscle strength through muscle tension enhance osteocalcin's secretion? Well. Muscle strength through muscle tension. You know, like doing very specific muscle. The answer to that is interleukin-6 coming out of muscle increase with muscle strength exercise, type of exercise. Do you know that, Fabio, if interleukin-6 increase with this sort of exercise?
The IL-6 secretion is dependent the time, the duration of exercise.
Mm-hmm.
How long exercise is increase the IL-6.
But
endurance training, the marathon increase more.
A lot more. So
Yeah.
I would say that I don't think that it would change much with anaerobic exercise. I don't think this thing from what I read is it's mostly endurance exercise. Nutrient utilization, and ATP production, not really anaerobic. For instance, the receptors is more found in oxidative than in glycolytic fibers. So it's more for aerobic type of exercise than glycolytic type of exercise. That's what I think.
And also
Any adverse effect when injecting osteocalcin? Well, thank you for the question, Josefina Bertoli.
Yeah.
Josefina from Argentina.
From Argentina. All right. Yeah. Okay. They inject osteocalcin in mice only in mice, and it doesn't seem to have much effect. Probably it's a question of what's the dose you give, how much osteocalcin you give, but they did not report any effect. I did not do that myself. So, like you inject anything, you have to make sure you have the right amount, and I don't think there was adverse effect in mice. In mice.
Jean-Marc, this guy, I think that you also don't know who he
Gustavo.
is.
Oh, Gustavo. Hey, nice to see you, Gustavo. Hey, and congratulations for your wedding. I heard you get married, since you came to Canada, discover your wife, and then said after Canada you get married. Good. Does high intensity exercise have any effect on the osteocalcin release? Ooh, it must have a little. It must have, but not in those studies that it's not being looked at. Those people working on this are not exercise type of people. They're not coming. Those studies and nothing coming from kinesiology department and so on. So they use classical type of exercise, with the mice. But I would say that high intensity. Well, I think I had this question before. I think it would have an effect. So, but you have to combine high intensity with long duration exercise. You have to have a mix. But it would certainly have an effect, on. I would even suggest that,
Mm-hmm. Here we also have one more question from Yuri. He's a post-graduation too.
Mm-hmm. Is there a difference between the type of carboxylated and decarboxylated osteocalcin in crosstalk with muscle? Yes, there is a difference. Yeah, there's a difference. It's specific to the mice, and the other is specific to humans. And probably I don't know, in the mice, if you don't have this carboxylated osteocalcin, it wouldn't be as much active. I wouldn't say it's not active at all, but it would not be as much active. And in human, if you don't have if you don't have the carboxylated, then it disappears very rapidly. So 90 minutes half-life, something like this. So there is a difference. And probably it would still be active. It would still be active, but not as much. That's why people, as I said, they try to carboxylated osteocalcin and inject it in human. And they will last longer, but maybe not as effective. Molecule, it depends. Where the carboxylated is on the molecule, it's the active site or not. But yeah, it makes a little difference.
I think that all questions finish here.
Okay.
Or if someone wanna send some more, but I think that's it was good questions that we could discuss.
Very interesting. Very interesting. Yeah. Good. So we have to say goodbye. Is that what it is? We're at the end of it.
Thank you, Jean-Marc.
Hey, obrigado to all of you. Good luck in your work. And stay healthy. Stay healthy as much as possible. Also, everything in Brazil is difficult. it's difficult everywhere.
Yeah.
You know?
Yeah.
Yeah,
It's difficult
exactly.
everywhere, but we have to be all patient and stay calm. You know, we'll go through this. It will take some time, but things will come back to normal in very soon,
For sure.
hopefully, as much as possible. But we have to pay attention now. All right.
I would like to say for everyone here that I'm putting people who did the inscription. I'm putting now the forms again to have the subscription and to have the certification to participate of this meeting. So everyone, please fill these forms. All of students who did the inscriptions for the meeting. And I would like to finish this presentation saying thank you so much for presence of all of you. We had 100 with Jean-Marc knowledge, you know, it's easy to have attention from a lot of people. But I really would like to say thank you for Jean-Marc and the presence of everyone here. I appreciate your presentation, Jean-Marc, and now we have to prepare ourselves to the next presentation tomorrow and bring good questions like today. And for who did the inscriptions, we have also a fold in the link of inscription that has all papers that Jean-Marc sent, and also the Professor Ana Maria for tomorrow. So everyone who wanna read those papers too and discuss more deeply about about this presentation, just check it there, and that's it.
If you talk to your colleague Brazilian, you could talk in Portuguese, you know.
Okay, but it's English presentation.
Your English is so good now.
Eu sais aussi parce que j'ai pratiqué mon français, mais c'est un peu tout.
Wow! I'm amazed here. Wow! All right, guys. Take care. Huh?
Okay, thank you, Jean-Marc.
Okay, ciao.
Thank you,
Bye.
Obrigado.
Obrigado.
Obrigado to you. Ciao.